All right. Thanks so much for that introduction, Ben. I guess probably my logos need to be updated as we go through our merge here. So a lot of the talk today, I'm going to be talking about a particular platform that we've developed called Minecraft-seq. And I just want to quickly acknowledge the folks who've really done the work, in particular Yuri Baglienko, who's really been the driver behind this experimental approach, who's now at Cincinnati Children's. Imagine Ryan helped with the experimental component of it. Michelle and Aparna helped us develop the statistics. And we had a lot of help from Greg and David in terms of the CRISPR analysis. So it's been great to hear all the different perspectives on going from a variant to function. And you know, as Ben mentioned, I think a lot about autoimmune diseases, and I come at it from that angle. And I think as a lot of the talks alluded to today, you know, for a given locus, it's really critical to not just discover an association, but to find the function. And to do that, you really need to understand what the causal SNP is, and that's often a challenge because you have multiple SNPs associated, as you can see in this particular RA-associated locus. You want to understand the causal function of that SNP, and that's, you know, again, typically a non-coding regulatory function in most 90% of cases. Once you have that solved, figuring out the causal gene, and doing all of this requires us to have some sense of the causal cell state, especially because gene regulation is so cell state specific. So I sort of think about these problems as like very interlinked with each other. So it's a little bit like a Jenga tower. You really can't address one in isolation of the other three. And one of the things we've been thinking a lot about in the lab, and as many folks here have been thinking about, is, you know, how can we exploit single cell resolution data to define causal variation. And on the right here, you have a UMAP of T cells, the cell type I kind of think the most about in the context of autoimmunity. It's possible to examine precise cell states with single cell data. A lot of times you have a lot of power because every cell can be its own measurement, and so there's some utility there. And multimodal data can be really useful, thinking about ATAC data, RNA data together, thinking about which cells have a particular variant or perturbation in them. But the statistics are often complicated, and sometimes there are a lot of technical challenges. And what I'm going to be talking about a lot today is really trying to look at DNA level data, such as targeted DNA sequencing and RNA together. So trying to obtain that data in an individual cell has been one of the things we've been trying to do. I'm going to allude to three V2F activities, and I'm going to spend most of the time today on the third one. First, I'm just going to allude to a little bit of work that we've done trying to map cell types or cell states, specific EQTLs. And as we talked about, a lot of variants have cell type specific gene regulation. This is the IL-6R locus, which is associated with rheumatoid arthritis. You can see that there are, it looks like there's just one association on the far left, but if you take that SNP, you condition on it, you see a second association emerge. So you have really two independent SNPs driving the RA association in this locus, and they're independent of each other. And on the right, what you see is a splice QTL study in monocytes and an eQTL study in CD4 T cells, and you see that these variants both affect IL-6R expression or splice isoforms, but the two alleles are doing it differently in the two cell types. And to complicate matters, if you think about IL-6R specifically in T cells, as I mentioned, one of those variants are acting in T cells specifically, it's actually more complicated than that, right? IL-6R, you can see in this T cell UMAP, is really expressed in this subset of T cells in the upper left. These are T peripheral or T follicular helper cells. These are T cells that are very important for RA susceptibility, and that's where IL-6R is expressed. So it's a fair bet that this EQTL is really playing a role only in a subset of T cells. So we've thought a lot about this problem and thought about genetic variation and how it might influence a gene's expression differently in different cell states. So again, trying to take advantage of single cell data. In this study, we tried to come up with per cell betas for eQTLs. So you can imagine that there's some cell types or cell states within the same cell type, but there's no association. Others where there's a strong association. Using this kind of Poisson mixed effects model approach, we've been able to find interesting things like this. This is a MAF EQTL. And if you look at 3 different sort of samples, what you can see is that in the far left, there's really no association. In the middle, there's a weak association. On the far right, in the cytotoxic T cells, there's a really strong association. So we're able to find these kind of effects using population-based EQTL data. That's one activity that we've been doing. A second one is thinking about building high-resolution enhancer gene maps from RNA ATAC data. And this is work that Sari Sakawi has led. It's now in press. And then the idea here is that in many instances, we want to use ATAC-seq data to find gene regulatory elements. We want to find the ones that are specific to the cell type or cell state we're interested in. And we want to be able to link that to downstream genes. We've been looking at multimodal data. These are single-cell data sets with RNA and ATAC data in it together. One activity that many in the field have taken is to simply look for correlations between RNA level and open chromatin levels. And this is typically done in bulk data. And we had the idea that, again, you can take advantage of the fact that you have single-cell data to increase your power, again, if you are able to account for the sparsity accurately. And so Sari developed a bootstrapping method to do this accurately and to get better power. And so then the idea is that once you have a bunch of variants with plausible posterior probabilities, you can use this approach to not only find the regulatory element that's associated with a particular gene, and that's likely picking out the causal variant. You can also pick out the downstream gene. We looked at data from FinGen, UK Biobank, looked at the likely causal variants. We found tremendous enrichment in the scent-based defined peaks as opposed to bulk or ENCODE C-Cres and so on. So it has a pretty high predictive power. This is that same locus I showed you, and this is just a quick example of how it can be used. So here you see you have a whole bunch of variants. We're picking out one, which is sitting in a scent peak. And you can see that where the arrows are, that peak is really seen in T cells from RA inflammatory tissues. It's not seen in T cells from blood. So while we can get T cells very easily from blood, we do sometimes have to spend the effort to get it from the tissue. You also see it in the fibroblasts from RA tissue. Again, not a blood phenotype. But that allows you to link that variant to a gene. The gene's a little bit further away than you'd expect. It's not the closest gene. And that connects it to this gene RBPJ, which is important in Notch signaling, which is important in RA susceptibility. So I'm going to spend the rest of the talk talking about how we define the effective variants by gene editing at single-cell resolution. I think we would all agree in this room that there's a need for base pair resolution CRISPR editing. I think that's really important to validate the function of individual variants. So once you have a variant that you think is driving the regulatory phenomenon you have, you want to see if you can introduce that variant and reverse the phenotype. And single-cell resolution, again, we thought would be very powerful, but there's some really important challenges. The first is that when you edit a cell, you actually change the cell state. As I mentioned, a lot of these effects are cell state specific. So these editing conditions can induce non-specific environmental effects. A lot of the existing single-cell approaches work by sequencing the guides. But just because a guide got into a cell doesn't mean that you've actually edited the DNA. And once the sgRNA is incorporated into a cell, the edit may or may not happen. And particularly if you're interested in primary cells, as we are, primary T cells is, again, a big focus of our energy. Editing is particularly inefficient. So if you look at the circle, a lot of times we're doing this kind of work looking at pools of cells. But again, when you try to edit a pool of cells, only the small red circle of cells are edited. The pool might see some environmental effect. Some subset of them get the sgRNA incorporation. Some smaller subset actually get the edit that you want. So you lose a lot of power when you look in bulk populations or when you sequence sgRNAs. So we developed a protocol called Minecraft-seq, which is the multi-omic investigation of nucleotide editing with CRISPR by ADT flow cytometry and transcriptomic sequencing. It's very long. So Minecraft, if that means anything to any of you with kids of a certain age. That's what we went with. So we first edit the cells using something like a base editor or an alternative technology. We sort those individual cells into plates, and then we do targeted single-cell DNA sequencing where we're able to confirm the presence of the edit. And the real trick is we're also able to get RNA, genome-wide RNA data in those same cells. So getting both of those things to work together. We also happen to get flow cytometry, and we do site-seq, so we get a lot of surface marker data as well. And we can, because of the site-seq platform, we look at about 154 different surface marker proteins. And this is a schematic of the approach. So you have your cell, it's got the DNA in the middle there in the nucleus, it's got the RNA as well as the tagged antibody, and you're getting each of these different modalities of data and looking at it. And so the idea is when you want to understand an individual variant, you take your cells, you edit some of them, and then you have a non-targeted control. Normally we would compare, and prior to this approach, what we were doing in the lab is comparing just the edited pools to the unedited pools. And now what we do is you look at the control cells, which presumably stay with their native genotype, but the edited cells end up having a allelic series, right? And because it's single cell data, we can isolate specific cell types that we're interested in or cell states that we're interested in. And now on the right, we can find non-specific changes by comparing the control cells to the CRISPR cells. So that has nothing to do with genotype. That's just the experimental condition. But now within the edited pool, you can look for allele-specific effects and look for allelic changes, but only in the edited pool. So how well does it work? So I'm going to show you some pilot data from 3 different cell lines that we looked at. So here we had Jurkat, Dowdy, and HEC293T cells together. We put them together and sorted them into 2 384 well plates. And we sequenced this P10 locus, which the exon 7 region of P10, which has a known indel in Jurkat cells. And the data was very good. It gave us data that was pretty comparable to what you'd get out of a 10x run. About 58% of the RNA reads mapped to cells. 555 of the 768 cells passed stringent RNA-QC. We got about 50,000 UMIs per cell, capturing about 5,100 genes on average. And you can see the distribution on the right here. And when you cluster, you get these 3 very clean clusters capturing the original cell lines that you put in. You have clear marker genes. You see, for example, CD3D marking Jurkat. So you see a very clean separation of your different cells. Because you've got RNA and protein data, we can actually see stuff like this, where you've got CD4 expression on the bottom and CD40 ADT on the y-axis, and you see a pretty reasonable correlation modulo some sparsity between the protein and the RNA for this particular gene. And as I mentioned, we actually wanted to capture P10, so we sequenced exon 7 there. That was the targeted region we chose to sequence. We saw the reference allele, which has the gaps up at the top, but then we saw a subset of alleles that had a 38 base pair insertion, another subset of alleles that had a 9 base pair indel as well. And the allele frequencies are roughly similar when we sequence the region in bulk and also when we infer what the allelic frequency would be from the single cell data. And you can see the full heterogeneity there. So at the bottom, you see the reference allele. And you can see all the cells, except for 2 that have the reference allele, show up in Dowdy and HEC293T. So it gives you a sense that we're about 98% accurate. And the indels really only show up in the Jurkats, which, again, is this whole spectrum above it. And you can see that the 2 deletions show up in mixed and matched. They're always out of phase with each other. So there's actually 2 different alleles that are showing up in Jurkat. There's some heterogeneity there, which we found interesting. So the pilot showed us that we can apply Minecraft-seq to cell lines, and we're able to obtain high-quality RNA, surface marker data, and also DNA allele calls. And so the next test is, can we edit a causal allele? So we started with an EQTL, RPL8. This is a EQTL gene. We looked at it specifically in B cells. This is a well-known pan-tissue EQTL. We focused on B cell lines. We picked a particular variant, this RS2954658. It's likely the causal variant, as I'll show you in a second. And what we can do is we can use AB8 editors to edit the T alternative allele to the C reference allele. So this is like 3 different data sets from the Michigan EQTL browser. And you can see the variant that we picked there at the top. It's basically the strongest associated variant in these 3 different data sets of LCL blood and naive B cells. You calculate posterior probabilities, and you see that that same variant either comes up as the individual variant or the highest scoring posterior probability variant. So we went ahead for it. We assayed, again, about 700 or 800 cells. And at the bottom, you see the reference individual. So those are all the cells, the majority of the cells that we failed to edit, again, showing you how inefficient things can be. In the middle, you see the heterozygotes. That's a subset of about 194 cells. And then about 26 cells, you get both alleles in. So you see the allelic spectrum. So now if you look at the control versus CRISPR, if you were going to do this in pools, you'd be really disappointed, right? Because you'd find that the control and CRISPR edited cells have like roughly the same expression of RPL8. But now watch this. Let's look at those edited cells and break it down by genotype, TT, CT, and CC. We do a sort of negative binomial model, exactly like what we did for our EQTL type of studies. We have a genotype term, and we can calculate significance. And what you see is there's a 10 to the minus 6 association in just several hundred cells as you go from 0, 1, 2 C alleles with RPL8 expressions. You see a dramatic, statistically significant increase. You might be wondering whether this is specific or non-specific. So as I mentioned, we've got genes genome-wide. We can test about 5,000 different genes. So here's the Manhattan plot. You see the strong association for RPL8 for this variant. You see no association in any other gene throughout the genome. So we're able to really specifically identify the target of this particular EQTL. So just to summarize, we edited this RPL8 RS295 blah blah variant in Dowdy B cells with ABE8. We observed low efficiency, and hence bulk differences in the edited and control populations is minimal. But when we model allele dosages with a single cell model, we see significant differences. And the differences are only seen in RPL8 and no other gene, arguing for the ability to find very specific effects. All right, so that was a cell line. I promised you we'd work in primary human T cells. So these are, that's what I'm going to show you now. So we want to examine primary T cells specifically, and we picked a gene, PTPRC, which is really important in T cell signaling. It's really an important gene as T cells make a transition from naive to memory T cells or activated T cells. It undergoes an isoform change as it goes from naive to memory. And what we did was we edited a loss of function allele into this gene with the BE4 editor. So again, we saw a spectrum of different alleles. You see the reference allele, which is, again, just normal PTPRC. You see individuals with 1 or 2 edits in it, and then you see a few less desirable changes. And what you see when you look at this is, again, the majority of the cells are not edited, and a minority of cells get a single CT change or a double TT change. You can see in the controls, generally we don't see any edited cells, which of course makes sense because they have a non-targeted guide in it. And now if you look at the RNA levels, what you see is you see a reduction in expression as you go from 1 to 2 alleles. This is probably driven by nonsense-mediated RNA decay. But more strikingly, when you look at the protein levels, that's where you see a real difference. The individuals that have 2 of these loss of function alleles in TT, they have almost no protein expression of CD45, which is the gene encoded by PTPRC. Can we find loss of function trans effects, right? So we're breaking this pretty important gene. Maybe it has other effects in other genes. So we thought even with our kind of hundreds of cells, we might be able to find something. Here's an example of a gene that we did find. This is TNFRSF4 or OX40 or CD134, important marker of T cell activation. And what you see is that as you introduce those PTPRC loss of functions, this gene loses expression both at the RNA and at the protein level with a 10 to the minus 5 significance. So a pretty strong effect on this particular gene. 60 out of 544 genes have some kind of effect with an FDR of 0.2. You can see a really small number of genes have a very profound effect, but a larger number of them have more subtle effects. Protein is really interesting. Obviously, a lot of the proteins that we're staining for are T-cell activation markers, and you see that a lot of them are indeed affected as you introduce more copies of the loss of function allele. And you can do a pathway analysis of the genes, and as I mentioned, PTPRC is really important in activating T cells and making that naive to activated or memory transition. Indeed, we see enrichment in a pathway analysis of these genes that are downstream of this gene. Now, as I mentioned, sometimes you can see these kind of pooled effects. So on the bottom here, you see the association with the genotype. And again, you see TNFRSF4, PTPRC, strong association, like I told you about, because that's where we have the loss of function allele. On the y-axis, you see the association of control versus CRISPR. And I thought this was really interesting, because what you see is you see an interferon signature, right? So in the pool of edited cells, there's a environmental effect of reduced interferon. In the unedited cells, you have actually a higher interferon level. So if you were doing a pooled comparison, you might think interferon signature is important, but you'd be fooled. Because again, that reflects an environmental change that's indirect. So the effect of a PTPRC loss of function, just to summarize here, editing a loss of function PTPRC allele induces changes in T cells. Loss of PTPRC gene expression is definitely something you see due to nonsense-mediated RNA decay, but a profound loss of function of the protein, because it's a loss of function allele. RNA can't be used to make protein anymore. We talked about some trans effects, and we actually talked about how this affects the T cells at a large level, at the phenotype level. And we can distinguish non-specific interferon signatures from genotype-mediated activation signals. And that would be a limitation of a bulk assay. So finally, I'll talk about an IL-2RA locus. There's multiple variants in this. There's one particular variant that I'll talk a little bit about. It's been seen in type 1 diabetes, asthma, lupus, and rheumatoid arthritis. Previous studies have argued that it's likely to be causal. There's some studies where it's been shown to increase protein expression, others where it's been shown to decrease protein and gene expression. So we thought maybe there's a kind of a context-specific, and in fact it's known from other studies that there is a context-specific component to this particular variant. We investigated in primary T cells, but this time we actually polarized T cells into two different states, Th1 cells and Treg cells, to see if we saw the same effect in both. And this is the experiment. So we're getting blood from individuals. We're editing the DNA. We also have a control sgRNA, and then we're polarizing the cells into Th1 and Tregs. And then we're applying Minecraft-seq and seeing what happens. So this is the spectrum of different genotypes that we see. You can see we did two different runs. In both instances, we see sort of some representation of each of the different genotypes. And these cells are proliferating in some cases, right? Because these are T cells. The fact that you're polarizing them, you have to proliferate them. So you see that about half our cells are proliferating, half of them aren't. And these are the results, a lot more subtle than the other ones that I showed you. Obviously, we're working in primary cells. And what you see is if you look at proliferating or non-proliferating Th1 cells on the left, you don't see any change in IL-2RA gene expression. And on the right, CD25, which is the protein, you don't see any change in gene expression. But if you look at Tregs, you see an association with the T allele with increased IL-2RA expression, more strong in proliferating, not significant in non-proliferating at the gene level, but nominally significant at the protein level. So this, we're really excited about this protocol. We think it really gives us the ability to enable the investigation of individual variants. It's a single cell, we think single cell modeling of variants is well powered and can be highly specific. It's not limited by low efficiency editing. So when you have a particular CRISPR protocol which doesn't have a lot of efficiency in primary cells, that's not a limitation for us in the way that it often is. It's $3 per cell, which sounds like it's kind of cheap, but obviously as you do thousands of cells, the costs add up. The limitation is that it's not as high throughput as we'd like, because obviously for every locus that we have to go after, you need amplicons, you need targeted targeted DNA sequencing, and you need to get that up and going. So single-cell technologies offer a lot of power to define eQTLs, enhancer gene maps, and other V2F activities. We present Minecraft-seq, which is this multimodal or multi-omic assessment of RNA, DNA, and surface marker protein. And it's a powerful platform to look at individual variants. And I presented some examples for loss-of-function alleles, for common variant regulatory alleles, and a disease allele. And the major challenge that we're working hard on is figuring out how to get this to scale. But we do think that you get very accurate information out of it. And we plan to apply this to a broad range of primary cells. And we're open, obviously, for collaboration. And with that, I'd just like to thank our funding sources and of course, the lab. And with that, I'll take questions.
Thank you, Soumya. Questions for Soumya? Is Fortnite-seq next? Couldn't resist. So I guess there are a couple of different points of leverage over the efficiency question. Do you have thoughts on, like, if we make the editing more efficient, how much does that play out in terms of cost reduction? Like, is that where the real bottleneck is? Or is it actually just in the manufacturing and design of all the guides and all that kind of stuff?
Yeah, so there's a couple places where there are time and money costs, right? I mean, one is that it's a plate-based protocol. Right, so plate-based single-cell sequencing is a little bit expensive, but like I said, you could get 1,000 cells for $3,000 in terms of that. Where the time bottleneck is like designing. So you're doing targeted sequencing of part of a lysate of something you're getting out of a cell. So you have to be able to sequence an amplicon in a single cell. So it requires a little bit of time to kind of optimize the primers. I think you want your CRISPR, you want your editing to not be 0% efficient or 3% efficient. But I'd say for us, actually, super weirdly, a 20% or 30% efficiency is not a problem. Now, when you're doing a bulk assay, big problem, right? But actually, for us, that gives us the heterogeneity in our pool of cells that gives us the power to find what we want to find.
And that actually relates to one of the other follow-up questions that I always have about these kinds of experimental paradigms, which is, do you think the CRISPR guide and the targeting is changing the chromatin landscape that's having an impact on gene regulation? So when you look at the control guide versus no condition, do you see differences in expression patterns accordingly? And does that influence the interpretation of your results?
Yeah, great question. So is it not the allele itself, but is it the fact that something is messing with that region? Yeah, you're breaking stuff. So we probably would need some more cells to answer the question. But what we do see, Ben, is that there are cells where you didn't get the edit that you want. You got some other edit, right? So you can actually ask the question, is the presence of an edit that I don't want affecting expression, in a problematic way? There's another example I didn't show where, we introduced larger and larger and larger breaks in a regulatory region. And you see that as the break gets larger, the expression of the gene gets lower. So it's not just a non-specific change, it's that specific region.
All right, any other questions for Soumya? I mean, I can keep going, but we should maybe keep the session moving. Oh, go ahead, Jose.
I was struck, maybe I didn't see it, but in the box plot, I was struck at the variability in expression. So you have many, it's true that on average you do have an allelic specific effect, but you do have many cells that have levels of expression when mutated that is similar to the wild type, in the very same range. So it really tells you that just changing that single nucleotide may not really be enough. There's a lot more complexity going on that goes beyond that. So in terms of going from an in vitro assay to what is happening in a person who's lived 50 years, et cetera, there's so many leaps that makes me a little bit cautious.
Yeah. No, obviously we're looking at the most proximal thing, right, which is the effect of the variant on a gene. It gets really complicated. We have another study that we're doing where we're looking at the effect of variants on cell states, and it's pretty interesting because sometimes you see a variant that we know to be acting on a gene to be affecting a cell state, but the cell state isn't expressing the gene, right? So probably what's happening is the variant is expressing a gene in another cell, type or cell state that's then affecting, some different cell types. So there's obviously a lot of complexity there, and I think this is just a first step in that.
Super. Let's thank Soumya again.